Rick is an international authority in neuromuscular disease, as you know, all well know, and a decorated researcher nationally and internationally, most recently receiving the Researcher Award by the American Association of Neuromuscular Electrodiagnostic Medicine. So this is an interesting topic. We look forward to hear about a tale of not 4, but 5 INDs. And so stay seated and put on your seatbelts for a wonderful presentation. Thank you, Rick.
All right. Thank you, Mazen. Well, Happy New Year, and welcome to the first Neurology Grand Rounds at Neurology Neurosurgery Grand Rounds at KU Medical Center in 2014. And it's holiday time, so everyone thinks about Charles Dickens at holiday time, but not because of this quote, which is from A Tale of Two Cities. So everyone always remembers the first line from A Tale of Two Cities. It was the best of times, it was the worst of times. But then often people forget how tremendous the rest of the sentence was. It was the age of wisdom, it was the age of foolishness, it was the epoch of belief, it was the epoch of incredulity, it was a season of light, it was a season of darkness, it was a spring of hope, it was a winter of despair. We had everything before us and we had nothing before us. So that maybe can sum up the state of medical research right now. Sometimes things look pretty dark and you wonder where you're going to get your next grant dollar or where your next idea is going to come from or how we're going to continue the discovery enterprise at medical centers, which is what we're all here about. But there always then seems to be some light at the end of the tunnel, and we seem to pull out of it and are able to work toward our goal of improving patient health. So this is a tale of 5 INDs, which IND stands for Investigational New Drug Application, which our team at KU and our extended research network around the country has been involved in. And I thought I'd tie these talks together using the IND theme, because most people don't really know what an IND is. No one teaches you this in medical school, residency, fellowship. And so you just have to sort of learn about it on your own if you decide to take the step into this area where you're going to try a new drug on a patient population. And so I think it's useful knowledge to know about and to know how to get help if you want to do a study that involves an IND. And when do you need to do it? And then, but the rest of the talk is really about several rare neuromuscular diseases and some of the attempts that we are making to try to find effective and new therapies for our patients with these rare disabling diseases. So an investigational new drug application is actually a federal process, and it's the regulatory mechanism for new drug development. So when do you need to get an IND, which ultimately is a number, a federal number for your study? When do you need to get that? So the federal rule says that a sponsor, and when it says sponsor, that means you're the sponsor. If you decide to do a new drug study on a patient population that hasn't been studied before, you are the sponsor when you apply for the IND. It's not KU, actually, and it's not the drug company, it's you. Now, if the drug company is trying to do this for a brand new drug, then they are the sponsor, the drug company. But in our situation, it's us, the investigator. So a sponsor shall submit an IND to the FDA if the sponsor intends to conduct a clinical investigation with an investigational new drug that is subject to 312.2A. So that's what the regulation says. And you have to submit it unless you read the exempt criteria and you think that your study is exempt from requiring an IND. In addition, beyond the federal regulations, there are certain grants, certain federal grants, particularly FDA Orphan Products Division grants and some NIH grants, that absolutely require you to have an IND, even if you meet the exempt status. And that's why we've had to file INDs a number of times. So the issue is what gets you exempt for your study so you don't have to get a federal IND number. So these are the rules, and I've had a lot of help in paraphrasing these federal regulations from Laura Herbelin, our project manager on these studies, and Joanne Miller, our regulatory manager in the Research Institute. And they've helped more than just simplifying the slides. They actually help get these INDs done for us and do the work to get this to happen. What the rules say is you are IND exempt if you are not trying to get a change of indication or drug labeling. So most of the time, so that would apply. I mean, we as investigators at universities, or if you're in private practice doing a study, are really never trying to get labeling indication. That's the job of pharma. So there may be some exceptions to that, but I haven't come across them yet in my career. So maybe there will be a day when we are at the level where we can actually do a labeling indication if we have some sort of collaborative process with a drug development organization, but it's not happening now. So number 2 is no change in advertising. Well, that's usually the case too. We're not going to do new ads in JAMA or Neurology or Journal of Neurosurgery to change the indication for our drug. No change in the route of administration, dose, patient population, or increased risk. This is where the rubber hits the road, and this is where it's all in interpretation. If you think that you're using, there's a drug that's been used for something else, and now you want to use it, it's been used for disease A, and now you wanted to use it for disease B. But it's a possibility that the patient population that you're studying has a different risk level and is at increased risk, then you have to get an IND. That's all, and that has to be determined by you, the investigator. You can get help on that interpretation by the FDA, but the bottom line is you have to make that initial call. Then all studies have to be IRB approved. So that really is not the issue. But the big issue is number 3, is the patient population at risk. So there's these federal rules out there. And if you're ever getting ready to embark on a new study with the drug in one of your patient populations, you need to sit down with someone in our research institute who handles INDs and really think this through. Look at those and go through them one by one and decide if you want to apply for an IND number or if you want to apply for IND exempt status. So as an investigator, you can decide if you're exempt or not. You actually don't even have to send a letter in to ask the FDA if you're exempt. It's my understanding, and no one's ever told me I'm wrong on this, that you read the federal rules, and if you decide you're IND exempt and your IRB agrees that this study is IRB exempt, then you're good to go. Most of the time, and I would recommend that if you do think you want to be IND exempt, that you write the FDA with your idea and to Dr. Katz at the FDA, and they will get you a letter back telling you whether or not you're exempt or not, and whether or not you're good to go. Or they'll say, no, you're not exempt. You need to apply for an IND application, and we're going to critically review your protocol and weigh in on it. So if you go ahead and get you apply for an IND, and I'll tell you what that involves in just a minute. What are your roles and responsibilities? So again, you are the sponsor investigator. And it's always an individual, and there are certain requirements that you now have as the holder of that IND for studying that drug in that patient population. So you have to submit several federal forms to the FDA, and the main one is this 1571. And that is where you discuss your protocol, and you talk about some of the preclinical data that you usually have not have done. That's been done by someone else, and then you have to cross-file on the preclinical data with the pharmaceutical company that did that original work. And that's often the most difficult thing, to get a letter of support from the drug company to allowing you to cross-file on their preclinical work on that drug in another patient population usually. 1572s we all do. Any investigator involved in the study, if you're an investigator or a sub-I, you have to be on 1572. And then you have to post it on clinicaltrials.gov. So when you send in your application to get the IND in, to the FDA, the day that they receive it, a 30-day clock starts. And by federal law, they have 30 days to review the protocol and to decide, number one, is it exempt or not, if you're asking for exempt status, or number two, if it's not going to be exempt and they're going to issue you an IND number, if they're going to let the protocol go through as is, or if they're going to make changes, require you to make changes. And if you don't hear anything in 30 days, then you're ready to go with your study. So it's within that 30-day time period, they can call you at any time. It's usually during the day, but I've been called in the middle of a clinic before, where you get on the phone and it's the FDA and there's a room full of people there and they're ready to talk to you about your protocol, and that's actually how it usually works. You have no warning when they're going to call you, and then they ask you a few questions. And then they get back to you with whether or not they want to apply. They usually say you have to give more written information, or they'll get back with you saying you're ready to go or not, and we'll approve your IND. So if you don't hear anything within 30 days, then you're ready to go. In those federal applications forms that I mentioned, what goes in there is a cover letter, your investigational plan, which is a shortened version, usually the investigational brochure that you have to get from the manufacturer, the draft of consent forms, and then the all-important cross-reference letter from the manufacturer that did the preclinical study. This has really stopped some people from getting an IND, because if a drug company does not want this study done in another patient population, that it might increase their risk of finding other side effects from their drug, they could not allow you to cross-file on their IND. And that has happened. And so that's when you're hoping your IND exempt, and it doesn't make any difference. But if you're applying for an NIH or an FDA grant that absolutely requires an IND number, then you have to get that buy-in from the drug company. So after you get an IND number, then there are some annual updates that you have to do that you wouldn't have to do otherwise. So we all have our IRB requirements that we have to do with our local IRBs, but here you also have to then file an annual report to the FDA on your IND anniversary to give them a status update. Anywhere along the line, if you have any serious problems, serious adverse events, you have to let the FDA know after you've let your own IRB know. and if you have any changes in your study teams, you have to keep them in the loop. And so you have to keep this active until the study is long over. So that's a little bit about the nuts and bolts of INDs. Let me run you through quickly some of the IND projects that we've been on. And the first you've probably heard me talk about before is our involves the rare diseases non-dystrophic myotonias, which is one of the inherited muscle channelopathies. And these are disorders usually of the sodium or chloride channels in which patients have episodic weakness or myotonia. But for the purposes of this project, we were looking at myotonia and paramyotonia. And so we decided to look at the old drug, mexilotine, the old cardiac drug, in non-dystrophic myotonia. Now, we had to get an IND on this because we applied for an FDA Orphan Products Division grant, and in order to get that money, they required you to have an IND. Now, the preliminary work on this project had been funded through a number of sources at the NIH, through the Rare Disease Clinical Trials Network, and which funded our CINCH consortium, where we're investigating neurologic channelopathies. Dr. Griggs is the PI on that, and I was the co-PI. And we also had involvement with the Muscle Research Center in England and Dr. Hanna. But in order to do this phase 2 trial, we needed to get the FDA Orphan Products Division grant, which is only for rare diseases. We needed the IND. So we applied for the IND. I think this is one of them where they called me out in the middle of clinic, and I had to answer some responses about EKGs and other things that we were going to be doing in the protocol before they would give us the IND. But we finally got it, and that was our IND number here. And so it was a pretty simple trial design where patients got either mexilotine or placebo for 4 weeks, then they had a 1-week washout, and then they crossed over and had the other treatment for 4 weeks. We had 59 non-dystrophic myotonia patients. These are, again, pretty rare disease, hard patients to find even at big muscle centers. But this is an international study in Europe, Canada, and the United States. And our primary measurement of efficacy was this interactive voice response, where the patient just called in on a telephone and rated their symptoms of stiffness on a 0 to 9 scale every day for 9 weeks. We had some secondary outcome measures too, but that was the primary, the interactive voice response. And the results were just dramatic. Here, you can see where patients were first on mexilotine and then on placebo, and then when patients were first on placebo and then on mexilotine, and you can see when they're on mexilotine, their interactive voice response scores for stiffness were just dramatically decreased. Their symptoms of pain, weakness, tiredness were also decreased, and all of the secondary measures that we measured also were affected by this drug. So this was a dramatically positive study. It ended up getting published in JAMA, amazingly, which doesn't usually publish rare disease research. And the first author was our own Jeff Statlin. So Jeff started on this project when he was between medical school and internship. Before he started his internship and neurology residency here, he was our research assistant. And that's when we really first started designing the Myotonia project. And then he stayed involved as a resident. And then when he went on to Neuromuscular Research Fellowship in Rochester, I asked him if he would assemble all the data once we were done with the study. So he did all the heavy lifting to assemble this and put this paper together that for our team that ultimately got accepted at JAMA last year. So we were able to conclude that mexilotine did improve stiffness and pain significantly. It did have some GI side effects, which we predicted, so we think we need other alternative therapies. It's a hard drug to come by. Some pharmacies don't stock it anymore because cardiologists never use it. In the middle of the study, we ran out of mexiletine. It was a big problem trying to get it again. Our study was on hold for several months, and that was a problem. But we were able to show that we could do rare disease research in a big international study, and that you can use patient-reported outcome measures as a primary endpoint. So that was the first IND tail. The second IND tail begins with this front page from the article that we published out of this, and it's IVIG and myasthenia gravis. Gil Wolfe was the first author on this, and this was a negative trial. This is the first FDA Orphan Products Division grant that I got when I was in Dallas, and it was to see if IVIG would help in myasthenia gravis patients. And it was a large multi-center trial, and we had to get the IND because we got the FDA grant. And we had this very ambitious protocol to do 100 patients, some on steroids, some not. And after we had gotten about 15 patients into the study, there was a worldwide IVIG shortage, or at least there was in the United States. And we could not get IVIG anywhere. I'm still not sure what the cause of the shortage was, but you couldn't get it for clinical use, you couldn't get it for research use. It shut down not only this study, but also the GBS IVIG study that was NIH funded. And that study had to be stopped in pediatrics, and that study never was completed. So we could not show that IVIG worked in this 15 patients that were randomized. We actually had some patients do better on placebo infusions of albumin. And so there were a lot of lessons, though, we learned from this study in the 90s. Number one, it just points out once again what we already know. Underpowered research will often result in a negative result. And ultimately, the Canadians, Dr. Brill, Dr. Zinman, did an IVIG study in myasthenia that showed that IVIG is effective in myasthenia gravis, investigator-initiated study. So now we're trying to get pharmaceutical industry to jump in and do a labeling indication study because it's still off-label. Number two, you have to always be aware of placebo response. Number three, this should be on another line, some things we have no control over, and that's drug shortages. We just had no control over that. But you should always try to make lemonades out of lemons, and you should publish your negative data. Always publish your negative data. It's not embarrassing to do that. You have to get it out there. Usually, to put a trial like this together involves an enormous amount of work. And not only is it important just to publish the negative data for the sake of that data, but there are a lot of building steps that go into doing a clinical trial like this. And a lot of it has to do with scales that you develop. So when we first started doing the IVIG myasthenia project in the 90s, we went back and looked at the old QMG scale. We revised it. We did reliability testing. Laura Herblin was a key component of this. We had this paper published in the late 90s. Dr. Wolfe and Laura and I then developed the myasthenia gravis activities of daily living scale for that project. We published that as a separate paper, and that's now used in every myasthenia gravis study that's done now, and sometimes it's the primary endpoint. And as I added up, there are at least, there's probably more than this, 10 papers that stem directly from that negative data, that negative study of IVIG and myasthenia gravis, which we had to abort. And some of these papers are still coming out. So, and that leads to the third IND detail, which really does still stem from the first MG grant that we got through FDA Orphan Products Division, and we had to get an IND for, and that ultimately led to the methotrexate in myasthenia gravis study, which we are just finishing up on now here at KU in partnership with Children's Mercy. And I'll get to the Children's Mercy part in just a minute, since I know we have some of our children's neurologists here today. So in the wake of the negative trials of mycophenolate in myasthenia gravis, which were the two large mycophenolate MG studies were negative, that we and many others around the country were involved in, to everyone's surprise, So in the wake of that, we started looking around and saying, well, what else should we be looking at for myasthenia gravis? And Tony Amato and I thought we should be looking at methotrexate in myasthenia gravis. It's a drug that some neurologists are comfortable with when they treat dermatomyositis or polymyositis, but most neurologists don't think of it for myasthenia gravis. So we had this idea to look at it in myasthenia. Dr. Passnor is the co-PI on this project. And so it's a double-blind, placebo-controlled study over 12 months in which patients either get methotrexate or placebo, and they already have to be on prednisone. And it's to try to determine if methotrexate is safe and effective in that population of myasthenia gravis patients who are on prednisone. So we made a mistake when we sent in our FDA application for an IND on this one. And I didn't realize it till after I got this letter. So usually the FDA Orphan Products Division grants are due the second week of February. And so you have to get your IND application in by December 30th. And so we sent it in. It got there January 7th, because again, they have to have 30 days to review. And they said, Dr. Katz said, after reviewing the information contained in your submission, we have concluded that your study meets all their criteria for exemption for IND. So I said, I can't be exempted. I'm applying for a grant that has to have an IND number. But I didn't make that clear in my application to the FDA. So I made that mistake once. We've never made it again. So you have to tell them why you're asking for the IND number. So we had to scramble. We had to call the FDA up and said, no, we are applying for an OPD grant. We're submitting that February 2nd. They were very accommodating. They re-scrambled. They reviewed the protocol. I think they had us make some changes, and we were able to get our IND number and submit the grant. Because when you submit the FDA grant, you have to have an IND number on the face page of the grant application. So we were able to skate by that mistake. So this study is just finished. So in this study, we had to find 50 myasthenia gravis patients who are acetylcholine receptor antibody positive who are on steroids. This slide has a slight mistake, and I think I'll change it before I post it. These patients actually, we struggled with this terminology. They actually are not prednisone dependent. They're MG patients on prednisone. None of us have ever been able to determine what prednisone or steroid dependency is in any of our neuromuscular diseases, and that's a topic for another group discussion with among neuromuscular people. But these are patients who could have just been put on steroids in the last few months. But then you put them on methotrexate or placebo, and you try to see patients on methotrexate, can their steroids be tapered to a lower dose than patients on placebo? That's the idea. We decided to use the prednisone area under the curve as our primary endpoint between months 3 and 12. And there were a lot of reasons why we made that decision. I won't go into them now. We had a number of secondary endpoints. These were a lot of the scales that we developed on the first MG-IVIG trial, and then some scales that Ted Burns in Virginia developed. We started with, I think, only 6 or 7 or 8 sites, and recruitment was very slow. And we kept having to add sites, and we finally got up to 20 sites. And it was not till we got University of Toronto and Dr. Brill on board that she put us over the hump, and she actually enrolled 8 patients right at the end. Otherwise, I don't know if we would have ever finished this trial because we were really out of the funding time for the grant. It just shows, again, how difficult it is to recruit patients for a myasthenia gravis trial. So after I actually wrote the grant and got it funded, while we were working on the CTSA grant, and I started talking to a lot of people at Children's Mercy more, I found out that our rheumatology colleagues over there, along with Steve Leader and also some folks in Lawrence, we're working on a new polyglutamation assay. for people on methotrexate. And this is just totally out of the neurologist's knowledge range. Maybe Dr. Murphy, you and Dr. Lipashon know about this, but as a neurologist, I'd never heard of it. Rheumatologists really believe that how much methotrexate is more effective the more the drug is polyglutamated in your system. And you can measure that in an assay, how much the methotrexate is polyglutamated. Apparently the rheumatologists have known this for a while. And the children's folks with Lawrence people had developed a new polyglutamation assay. So we decided to do this very interesting personalized medicine biomarker approach in this study. So all 58 patients all 50 patients that went in the study, at the end of it, they had a polyglutamation test done. So we will be able to correlate if that's true or not in the myasthenia gravis population. So it is 58 patients. So we had our 58 patients screened, and then we ultimately enrolled 51. We had 6 screen failures, but we had enough to do this study, and you can see the reasons for the dropouts. The last patient did finish in 2013, and so now the data is being analyzed by Wendy Hay, our statistician here. Laura Herbelin and Mamatha Pastner are cleaning up the database. This is another way to maximize your publication output. So this is a paper that Mamatha had published in the Annals of New York Academy of Science about a year ago about the design of the trial and why we used the prednisone area under the curve dose and some of the other unique things about the trial, like the polyglutamation. So while you're doing a trial, there are opportunities to publish papers about your trial as a multiplier effect. So the one other myasthenia thing I'll mention, because people that know about myasthenia are aware of this study, the NeuroNext study group just approved another study, and that's rituximab for myasthenia gravis. Dr. Nowak at Yale is now the PI. Dr. Goldstein, Dr. Damaczki, and I are co-PIs on it. And we are going to see if rituximab works better than placebo for myasthenia gravis. 50 patients makes sense. We're going to do it in NeuroNext. Good project. Rituximab, that drug has some toxicity. You would think it would require an IND. So this is a letter Dr. Goldstein got back when he apparently asked for exemption status. So the FDA said that your rituximab protocol can be exempt. I still find this amazing. But so you don't ever know what the FDA has in mind. It's not a benign drug. You'd think that you would want to have an IND number for this, but the FDA said that this study didn't need it. So this rituximab study is going forward without an IND. So that's MG. So let's switch to ALS and our next IND tail. And this involves the big rasagiline project that some of you in the room are involved in. And it really is a two-part story. There's a 36-patient open-label trial, which we just finished, which currently does not have an IND number. And then there's a larger 80-patient placebo-controlled study, which we are just starting, that we got an FDA Orphan Products Division grant for. And so we have an IND number for that. And this project was originally Dr. Wang's idea to do this open-label study. Dr. Katz, Dr. Swerdlow, and I are the other PIs on this. And we do it through the Western ALS Study Group and the MSG Muscle Study Group. And so why rasagiline? So in addition to the MAO inhibitor effects that rasagiline has in Parkinson's disease, where it has a symptomatic benefit, it also works at a mitochondrial level in neuron cell cultures. And this has been known for quite a while. And so actually, out of the Teva labs, Dr. Wang found this article that rasagiline prolongs survival in SOD ALS mice. In Israel, Dr. Drury used rasagiline in her clinic population and wrote this up and thought that rasagiline may have an effect in ALS. Also, when we were designing this trial and when we were doing the 36-patient open-label trial, the R-pramipexole trial was still going on. Pramipexole is another drug for Parkinson's disease, which also works at the mitochondrial level, and this was being tried in ALS, R-pramipexole, and everyone was very hopeful this trial was going to be positive. It turns out, a year ago, we found out that trial was negative. So that sort of took a little of the wind out of our sails on this rasagiline project. If that trial would have been positive, I think that would have been even more interesting for rasagiline, but you always have to look at the glass is half full rather than half empty, and there may have been other reasons why the R-pramipexole study was negative that are being looked at now, but we are still interested in looking at whether or not rasagiline has an effect in ALS. The Europeans are also interested in this, and Dr. Ludolf in Germany and a large European group are looking at rasagiline in ALS. So Dr. Wang got funding from Teva through an investigator-initiated grant to do an open-label study. So we did a high-dose rasagiline, 2 milligrams a day. The typical dose for Parkinson's is 1 milligram a day. We gave everyone rasagiline. We did it at these sites in the Western ALS Study Group. We were the coordinating site, and we looked at the ALS-FRS, which is our typical rating scale, this 12-point scale. Here you can see in the first 4, we're giving some granularity about it, what it means. So this is how we track ALS in our clinics. And the aim was to determine whether or not rasagiline at this higher dose was safe in the patient population, and also to determine if we can affect mitochondrial function using biomarkers in this population, and whether or not it had an effect on the ALS-FRS. So with regard to the ALS-FRS, this was our results after the first 6 months. This study was open label, so the placebo group came from our prior ALS trials that we had been involved in the Walls group, in which we had patients on placebo. So we knew the slope of their ALS deterioration in that group. And so this was our rasagiline patients. And so it looked like at 6 months, we may have seen a little separation, but at the end of the day, when we finished the trial at 12 months, our rasagiline ALS-FRS slope was 1.18 decline per month, which is basically essentially the same that we saw in our prior ALS studies. So we did not hit a home run with the ALS-FRS scale. Where we did find very interesting results was in the biomarkers. So while we were designing this study, I went to Dr. Swerdlow, our local mitochondrial expert, to ask him, is there anything that we can measure that can measure mitochondrial function? And so he started thinking about this, and he put together a series of 5 assays that we were able to use on blood tests in our ALS patients before and after they got rasagiline. And these basically measured, the first 2 measured mitochondrial membrane potentials. The second was a lymphocyte apoptosis marker. The third was a measurement of oxidative stress in the blood, this ORAC assay. And the fifth was this BCL2 Bax ratio that you can measure in the blood, which is sort of the life-death ratio that the oncologists use, but they like to see it go in the other direction, and we want to see the cells live longer. And so this is what we found in these assays. Both mitochondrial membrane potential assays, we showed at 6 and then much more 12 months, the mitochondrial membrane potentials became more hyperpolarized on rasagiline. The apoptosis level, lymphocyte apoptosis, actually went down, and this was statistically significant on this assay. And then the BCL2 Bax ratio dramatically went up. The life-death ratio improved in the direction that we wanted it to go. And then the the ORAC oxidative stress capacity also went up a bit over 12 months. So this is really the first time that anyone was able to, at the beginning to use target engagement endpoints as biomarkers in an ALS study to see if we have any effect on tissue that made some scientific sense. And so we finished this study now, and Zach Mackey was our year 1-2 medical student who got involved in this project as a medical student in a summer research. And he became part of the ALS research team. He stayed way involved for the last 2 years. He's presented this data now at the American Academy of Neurology, and then most recently at the International ALS MND meeting in Milan. He and I both went there, and where he presented this poster, and he got a commendation award on the poster. And so he just put together the manuscript, and he'll be first author on this paper when it goes in next week. And so you can never start too early on these projects. So we think that there is scientific rationale to study rasagiline in ALS, even though we did not hit a home run on the ALS-FRS in this small open-label pilot study, and that we might be demonstrating mitochondrial target engagement. Since we didn't see a big effect on the ALS-FRS, we actually went to our CTSA ethics committee to get a consultation, and we were told that as long as we fully disclose what we found in our open-label study, it's reasonable to continue to do the placebo-controlled study, and so that's what we're doing. And so we are now just enrolled the first few patients here in Kansas in this new 80-patient placebo-controlled study. We're using the same Swerdlow mitochondrial biomarkers. Now we're adding in some urine oxidative stress biomarkers for Columbia, brain glutathione MRS biomarkers here at Hoagland and in Columbia, New York, and TDP-43 and platelets we're measuring by Dr. Agbas at KCUMB in all the patients as another biomarker. So that study now will take about probably 6 to 8 months to enroll and then a year to complete, so you'll hear more about that later. And so then the final IND detail that I wanted to quickly run by you is a project of arimoclomol in inclusion body myositis. So Dr. Domachky is the PI on this project. The co-PIs are Dr. Hannah in England and myself, and Dr. Pedro Machado and Linda Greensmith in England. This has been a wonderful collaborative project between University of Kansas Department of Neurology and Queen Square in London, which is the institute that Dr. Hannah leads. So you all know about IBM, and this is a graphic that Dr. Domachky put together demonstrating some of the areas of muscle weakness in IBM, which you're all familiar with: the finger flexor weakness, the knee extensor weakness, and then inflammation surrounding muscle fibers with vacuoles on trichrome and on H&E. And we all know that IBM, despite this inflammation, does not respond to prednisone or any other sort of immunosuppressive treatment. So we've been looking around for drugs to use for IBM for some time. And arimoclomol came our way through the ALS world. It's a drug that increases heat shock proteins in the muscles, HSP70, HSP90, and others. And it interacts with mitochondria. And it is thought that by increasing HSP70 and 90, it actually slows down protein misfolding. And protein misfolding is one of the problems in inclusion body myositis, as it may be in ALS. And so the ALS world first got a hold of this compound, and we were involved in this project, Dr. McVeigh, as you recall, This was published in Muscle and Nerve in 2008. Dr. Zukovitz was the PI from Mass General, where we were able to show that arimoclomol was tolerated in ALS patients, but it was a short safety trial. It wasn't an efficacy trial. And for a lot of reasons that I'm not certain of, that drug just never went forward in ALS. There were some regulatory reasons, but there were some business reasons too. The company that first made this compound decided to sell it to another company. Before that happened, when I was at one of the original ALS investigator meetings, I was listening about the story on this drug, and I thought we should try this drug in IBM. So I approached the company who initially owned the product about getting some of their compound that we can try in inclusion body myositis patients. And this is a schematic on how arimoclomol can potentially reverse the abnormal protein misfolding that can occur in a number of our neurodegenerative diseases. So the company actually liked the proposal, the investigator-initiated proposal, and that we submitted to them. And it turns out that Dr. Hanna's team in England had already been working on arimoclomol in vitro in cell culture models that replicated IBM, and they had wanted to launch on a clinical trial too. So we had applied for an IND, we had obtained it because this is a brand new investigational drug. There was no way we were exempt because this drug is not used for anything yet. It's completely investigational, and so we had to get an IND number, and we did that, and then we had already been working with Dr. Hanna closely on our CINCH project. And so we decided to join our teams together, and instead of doing a 12-patient study that we were going to do just in Kansas, we did a 24-patient study in Kansas and London. So the KU, or the Cuckoo study, or something like that. And so we have finished that now. And it was a placebo-controlled trial, randomized 2-to-1. When we applied to the FDA, I applied to give this drug for a year. The FDA, when we applied for our IND number, would not let us because at the time there was only safety data for 3 months on ALS patients. So they would only let us give drug to the patient for 4 months. But we sort of wisely decided to still follow the patients every month for a total of 12 months to get endpoint measurement data after they got off the drug or placebo. And so that's what we did. And our primary measurement of efficacy was the adverse event reported and labs, but we looked at muscle strength, doing quantitative muscle strength testing, manual muscle strength testing, the IBM FRS, which our team developed, and also muscle biopsy, and looking at HSP70 levels. So Dr. Jackson and I and members of the muscle study group developed this IBM functional rating scale in the 90s, actually, when Dr. Tawil was doing the beta interferon in IBM study, which was negative trials again, but a good result of this trial was that we developed this outcome scale. And so we utilized this in our current arimoclomol study. And so we did have 8 treatment-related adverse events in the placebo group and 14 treatment-related adverse events in the arimoclomol group, but so it was slightly more in the active group. The most common was GI. The one serious adverse event did not lead to drug discontinuation. And some of the interesting results is that despite the fact that the patients were only on the drug for 4 months, at least in this small sample group of 24 patients, it looks like the IBM FRS did not worsen as much in the active group, blue, versus the placebo group. Having said that, at the beginning, they weren't exactly equal either. And so this clearly needs to be, we need to look at this further in a larger study. But it is suggestive that the drug may have had some effect. And not only in the IBM FRS, but also in the muscle strength testing, there was a hint that there might have been an effect. The disappointing thing was that we did muscle biopsies on all these patients, and we did not see the HSP70 level go up in any of the patients. When you look at the mean of the HSP70 levels, we did not see an increase with arimoclomol. So our conclusion from the arimoclomol for IBM is that it was safe and was well tolerated. It may slow deterioration, but it's still way too early to tell. It's a pilot study. We disappointingly did not see any change in the HSP70 levels. If we would have seen a change in the HSP70 levels plus this, I think that would have been really exciting in order to do a follow-up study. But we're still excited enough based on some of the clinical measures that we saw. And we think that this pilot study does support further research in arimoclomol for IBM. So we actually submitted this, a larger study to look at 150 patients to NeuroNext. NeuroNext decided they did not want to fund an IBM study. That was disappointing. And so one of my mottos in clinical research is never give up. So Dr. Domachky submitted it then to FDA OPD a year ago, and he actually got a pretty good score. And so it's been resubmitted in October, and we should get our next score back in February. And I think we're close enough that we're probably going to get this thing funded. And so hopefully we'll start the next arimoclomol IBM study, 150 patients at probably 20 sites or so. I don't know how many sites you've finally decided. We haven't quite figured it out yet, but we'll need multiple sites to get 150 IBM patients around the world. And that will start hopefully next year. So that's my tale of 5 INDs. And I guess there are a lot of lessons learned that you can take from this story of 5 IND tales. So first, just about INDs in general. And neuromuscular disease. I mean, I think these projects do show that investigator-initiated research in neuromuscular disease is viable. It's very doable, and you can do it with novel drugs like arimoclomol. You can do it with repurposed drugs, and I think this is a major component of of the discovery journey and why we are in academic medicine. I mean, these are the sort of studies that whatever field you're in, whether or not you're in MS or Alzheimer's or Parkinson's or stroke, you should always be thinking, if you're going to stay in academics, what can I contribute to the knowledge base in my field, and what questions can I ask that will take the field to the next step? These are not huge leaps; they're all small incremental leaps, but they all add up ultimately to advancing knowledge. As you can see, all of these studies involve multiple team members from multiple sites. They're huge operations to do, and it takes a lot of active components and to carry one of these projects off. The other message I would say is get involved early. So I actually wanted to emphasize the fact that Jeff Statlin was the PI on the JAMA article, and Zach Mackey is the PI on the Rasagilene 36 paper that we're going to send to a very prestigious journal. We'll see how far it gets. But I think if you get involved early with one of these research projects, we want to make sure that our junior investigators are well recognized in this discovery process, and because we want you to continue in your career in clinical research. On sort of a wonky end, some but not all drug trials require an IND. The IND rules are complex. You really need to work with your local regulatory and team sponsor. Our liaison here is Joanne Miller, who's in the back, and we could not do any of this without her. Laura and I and Mazen rely on her every day to try to do this, because not only does she have to get our IND stuff done, but she has to make sure that all of our sites around the country are compliant too, so that I won't end up in Leavenworth one day, or Dr. Domachky won't end up in Leavenworth prison. So, and so you can't really do this without someone that knows a lot about IND regulatory work. And even when I think that a project should absolutely have an IND, the FDA will turn around and say, no, you don't need one. So you just never know, but you have to know how to do the paperwork and send it in and find out what the FDA wants. Clinical endpoint scales are important. I mean, they're really the foundation of doing clinical research in neurology for sure, and I think it's true in every field. And that's actually where your publications, your first round of publications are always in case reports and case series. Your second round of publications should be in endpoint scales. And then that builds to leading to actually being able to do these complex clinical trials. What I'm finding now late in my career is biomarker endpoints may be even more important than the clinical endpoints, especially in an otherwise negative or underpowered study. And this is clearly what the FDA wants to see, what the NIH wants to see now. They want to see more and more and more biomarkers. So every time we do a clinical trial now, we try to find some biomarker that we can add on to this. And on the Rasagine 80 trial, we piled on as many biomarkers studies as we could think of and that we could afford, actually more than we can afford. So the way I'm thinking of this now is you've seen some of these studies where there was a hint that the clinical was positive, but the biomarker was negative. You've seen some where the biomarker was negative, but the clinical biomarker was positive, but the clinical was negative, or vice versa. And I think if either one is positive, that should actually get you interested in moving the process forward with that investigational drug for that disease. If both are flat out negative, then I think you really have to go back to the drawing board and rethink whether or not you should be studying that drug for that disease. The other important message to the young people here who are just starting out in academic neuroscience is write. You know, all the stages along the discovery process, there's always something that you should be writing up, and I think you can sort of gather that from this talk. And then, again, to paraphrase Charles Dickens once again at holiday time, sometimes it does seem like the worst of times, but it is also the best of times too. And there are a lot of opportunities out there for doing clinical trials. There's a lot of teams to coordinate with here at KU and around the country and around the world in your chosen field of investigation. So I think I will stop there, and thank you very much. Any last-minute questions for the first grand rounds of the year? All right, okay, we'll let you all go then. Thanks a lot.